Hi everyone. This is Vessels lecture. You guys are doing Vessels in lab this week. Not even sure if you'll watch this video until after your test. I want to remind you that you have a test on Thursday and or Friday. I've got it set for a 48-hour window in which you can take it. It's all in the announcements. I want you to go look at the announcements. It will answer a lot of your questions. Whatever questions are not in the announcements, ask them in the discussions so that everybody can get the benefit of the question and question and answer. It's going to be on Canvas, by the way. You have 2 take-home question assignments worth 8-ish points apiece if you do a good job on them. You need to be prepared to upload those when you take your exam on Thursday or Friday. And as far as I know, what I've tested is that Word documents are uploadable and almost any kind of image is uploadable. So you can handwrite it if you want and upload it, or type it in if you want and upload it. That's the first 2 questions on your exam will be to upload those answers. Okay, so I want to do vessels. I'm going to go more quickly than I normally would through this first part, and then we'll get into the specific vessels that you guys are also learning in lab. First, I want to show you this super awesome photo. I don't know how many of you guys have been to Bodies or Body Worlds. There is a plastination technique that you can do in which you inject latex into the vessels and then remove all of the other tissue. And this is just the head, showing you exactly how many capillaries are within a region of the body. Of course, the head has really, really good blood flow. So collectively in about a 150-pound human body, there are about 60,000 miles of blood vessels. This is not quite as beautiful, but this is vessels of the whole body. Most of those vessels are, of course, capillaries. So I just want to talk about the structure of vessels in general. And this is the stuff I'm going to go more quickly through than I generally would. The structure of vessels in general is that basically they have an inner layer, a middle layer, and an outer layer. Each of those layers is referred to as a tunic, like a coat. So if we go from the inside to the outside, we have the tunica intima, which is epithelial tissue, really, really slippery, so you don't accidentally catch a platelet. It should be continuous with the endocardium and the heart. It is going to have deep to that a layer of loose areolar connective tissue just under the tunica intima. It's usually got some elasticity to it. But if you are in an artery versus a vein, what you are going to have is an extra layer of elastic. So do you see this? It's called the internal elastic lamina. Lamina means layer. And what that is an extra layer of elastic connective tissue that is deep to the, sorry, I got allergies today, so I'm a little bit dingy. It's deep to the loose connective tissue that's underneath the epithelial tissue. And then please notice that a vein does not have that elastic layer. Veins do not need to be as elastic as arteries because they receive blood in trickles and arteries receive blood in squirts. And then the next thing we get to is the tunica media, and the tunica media is the middle layer, and it's primarily composed of smooth muscle tissue. And the smooth muscle tissue is going to be much thicker in an artery than it is in a vein. It's thicker as much for maintaining the structural integrity of the vessel as it is for vasoconstriction and vasodilation. So arteries have really thick walls compared to veins because the pressure in an artery is much, much higher. So the tunica media And the tunica media is also elastic, but then the tunica media is also going to have another layer of elastic connective tissue superficial to the tunica media in an artery, but not in a vein. So let's talk about how this smooth muscle in an artery or a vein could cause constriction or dilation. Because if this is the vessel, the smooth muscle is arranged circularly. What will happen is if you stimulate that smooth muscle to contract, it will cause constriction. And if you relax that smooth muscle, it will allow for dilation. So contraction of the smooth muscle will decrease the size of the lumen, and that's called vasoconstriction or just constriction. Okay. And then the outer layer is called the tunica externa, or sometimes the tunica adventitia. And it has collagen fibers, elastic fibers. It's usually woven into, vessels are usually woven into the tissues around them. So if you ever noticed vessels in the cadavers and labs, the reason that they were separated is because we separated them so that you could see them better. Generally speaking, they're kind of woven into the connective tissue of like a muscle as they go by the muscle. So they kind of blend in. All right, so let's see. Now let's just do a little comparison contrast between the types of vessels. What's the difference between an artery and a vein, both structurally and functionally? So if we look at arteries, arteries have much thicker walls than veins. And they're also sometimes, they're sometimes called efferent vessels because they carry blood away from the heart, efferent exit. And as far as what arteries carry, hopefully you guys remember, arteries carry oxygenated blood in what circuit? What circuit, systemic or pulmonary, do arteries have oxygenated blood? Pause. That is the systemic circuit. And they carry deoxygenated blood in the pulmonary circuit. Okay? And the vessel wall is thicker and stronger because it needs to be. The pressure is way higher in an artery. And their walls are more elastic because of these extra elastic, the internal and external elastic lamina. Arteries are more involved with muscular contraction just for stability. And then arterioles specifically do a lot of constriction and dilation. So let's look at a cross section here of an artery versus a vein and make sure that you can tell the difference between an artery and a vein. Most people tend to think before they get into this that arteries actually have a bigger lumen, but that's not generally true. Arteries generally have a thicker wall. They're easier to find when you're looking at a cadaver or even a living person because the thick wall makes them stand up and hold their shape, whereas this thin wall tends to make veins collapse. So arteries are easier to find, they're easier to palpate, they're kind of bouncy when you touch them. Most organs don't just have a single artery going to it, they usually have two arteries with a little route between them. What am I thinking of? An alternate route of circulation, and that alternate route of circulation is called an anastomosis. And the redundant circulation of two arteries going to the same organ, that's called collateral circulation. It's kind of a just in case, right? If there are two arteries going to an organ and one of them gets broken, then you still have that redundant or collateral circulation. Okay, so let's talk about the kinds of arteries that exist, and we'll go through this relatively quickly. So as you move away from the heart, the arteries are going to get smaller, of course, but they are also going to sort of act slightly differently. The artery that's closest to the heart, if we're looking at systemic circulation, would of course be the aorta. And the aorta has got a really big diameter, both in the lumen and the size of the wall. What it has is it's really, really elastic because the heart pumps into the aorta in big squirts, big boluses of about a 1/4 cup per squirt And so it receives this big bolus of blood and it has to kind of snap back. Those are the largest in diameter, is the elastic or conducting arteries. They don't do much vasoconstriction and vasodilation, even though they have tons of muscle in the wall. The muscle is mostly for structural stability, because can you imagine that you would really not want your aorta to constrict, because then the blood would actually get trapped in the heart. So they're more active in elasticity than in contractility. And then as you move away from the heart, you get to most of the named arteries, like the brachial artery or the femoral artery or the renal arteries, or any of the named arteries that you can think of, except for the ones that are right next to the heart, are called the muscular arteries, or sometimes distributing arteries. They're medium-sized or smaller size. You can see them. And they have proportionally more muscle for the size of the artery. They don't have more muscle, but they have more proportional muscle, and fewer elastic fibers for the size of the artery. And when you get here, they're more active in vasoconstriction and vasodilation. They're not just like holding structural stability. And then the ones that have the least elasticity and are the most reactive for vasoconstriction and vasodilation are arterioles, because arterioles are really the gatekeepers to capillaries. So when an arteriole constricts before a capillary bed, it decreases blood flow. When an arteriole dilates before a capillary bed, it increases blood flow. So when we get down to the arterial level, luckily you don't have to name them individually. First of all, they're really difficult to see. They're about 0.3 of a millimeter, and millimeters are tiny. And then second, they're really variable between people. So even if you named my arterioles, they wouldn't be similar enough to yours to be really useful. So they don't have, they've got mostly muscle in their tunica media, and they do a lot of vasoconstriction and vasodilation. That's kind of their thing. And then they're going to lead to capillaries. And capillaries break all the rules that we learned before about the tunics, because really all a capillary is just simple squamous epithelium, which we call endothelium when it was part of the cardiovascular system, and then just a basement membrane underneath that. They don't have any connective tissue in their wall, which is great for gas exchange, nutrient exchange, and waste exchange, but it's not very protective. So they don't have all of this protection in their wall. That means you need to protect them, if necessary, with the arterioles that are leading to them. Okay, and then we'll get to the types of capillaries that exist. So let's talk about what capillaries are for. Capillaries are primarily for exchange. They are microscopic. They're so tiny. Let me show you what one looks like. Oh, shoot, I forgot to bring that picture up. They are large enough in diameter just to allow a single red blood cell to slip through, single file, at least the smallest capillaries are. And they're the only vessel in this whole story, all of it, that allows a significant amount of gas, nutrient, and waste exchange. So really, the rest of this is plumbing to serve this, because without gas, nutrient, and waste exchange, the tissues don't get oxygen and nutrients and can't do cellular respiration, which means you're going to croak anyway. So capillaries are, like we saw in this figure all the way back here, they're found in close proximity to pretty much every single cell in the body. Although a few places you don't have a lot of them, like in the epidermis, and that's okay because it's thin enough that it gets nutrients and oxygen from below. You don't have very many of them in like hyaline cartilage, that's why it doesn't heal very well. The cornea and the lens of your eye, but most places in the body, they are within like millimeters of every cell that you could possibly run into. And so what do they do as far as flow? As far as flow, they are primarily really connecting an arterial, which is just a small artery, to a venule. They're going to be really branched because it gives surface area and plenty of time for exchange. And they are going to be higher pressure at the arterial end than they are at the venous end, because they have at least some level of porosity, and so some fluid gets pushed out. So you lose volume and therefore pressure as you go through. And then, of course, the ones at the arterial end are also just closer to the pump. So at this end, the arterial end of a capillary bed, it's going to be higher pressure, and at this end it's going to be lower pressure. So you lose water and solutes via filtration. Filtration is high pressure to low pressure movement of water and small solutes. So the venous end of a capillary bed is much lower pressure. It loses water, but about 90% of the water lost by filtration is pulled back in by osmosis, because you don't lose these big solutes that stay in the blood plasma, and they pull the fluid back in by osmosis. So you get turnover of interstitial fluid because of the filtration out and the osmosis back in. Okay, so let's look at the structure of a capillary. It's really just, let's see, it's really just a single layer of epithelial tissue called endothelium. It's simple squamous epithelium, and then a basement membrane underneath it. But there's some variability in the types of capillaries that exist. Some capillaries, by the way, have what we call precapillary sphincters. So I just want you to imagine that here's the arterial end and here's the venous end. There isn't smooth muscle everywhere through a capillary bed, but there are these little sphincters of smooth muscle. I want you to imagine that this is the capillary bed going through the smooth muscle in the wall of the uterus, okay? And then a tiger comes into the room. Do we want to wait for the blood to slowly percolate through all of these branches of the capillary while the tiger is trying to eat us? Or do we want to shut down blood flow through these extra little branches with these? These are called precapillary sphincters. And what they do is they force the blood through a thoroughfare channel, and so it can go back into circulation more quickly. So this is with them open, so no tiger, and this is with them closed, so tiger. So those are called precapillary sphincters, and they exist usually close to the arterial end of the capillary bed, but it's not like the capillary has smooth muscle everywhere, just at those locations. Okay, so let's talk about the three types of capillaries, and then we'll actually get into just do veins and then get into the vessels that you guys are learning in lab. Okay, so the three types of capillaries go from least porous, meaning most protective, to most porous as you move this direction. And so the most protective ones are these guys right here. They tend to have tight junctions between the cells, and they have a complete basement membrane. So generally speaking, with the tight junctions between the cell, if you want to get something from here to here, you can't go between, you have to go through, which gives a lot more protection. This is what you would find in the blood-brain barrier. The blood-brain barrier has even tighter ones than this continuous capillary. They're called continuous complete. Tight junctions just everywhere, because you don't want anything sneaking out of your bloodstream into your central nervous system tissue. So they have to cross through the cells. They have to get permission to come in. And then these guys right here have these little holes. They're actually little holes that just have cell membrane on them. All of the organelles and the cytoplasm is pushed to the side. So it's not just a big open hole, but it's like a double paned window. Either way, it actually increases transport. So these are called fenestrated capillaries. Fenestra means window. So they are found in places where there's a decent amount of exchange that's necessary. And then the last type of capillaries are these guys right here. These are called the sinusoid capillaries. And sinusoid capillaries have, this has fewer tight junctions than this. This has no real tight junctions to speak of. It's got big openings between the cells and then an incomplete basement membrane. This is found in locations in which you want to put whole big things in and out of the bloodstream, like the liver, monitors funky stuff as it goes through the liver. And what would happen is there would be a big white blood cell called a macrophage that was sitting up here, and as stuff went through the liver capillaries, the macrophage could go, oh, that looks weird, and reach in and, for instance, grab an E. coli that you accidentally ate on your poop spinach. Or you also find these in the bone marrow, because in the red bone marrow you make blood cells, and you need to get a whole cell from the bone marrow into the bloodstream, and you do it through these big holes. So continuous, fenestrated, and sinusoid capillaries. Okay, and then the capillaries are going to come together to form the smallest veins. The smallest veins are just called venules. Then we get to these medium-sized veins. They're just called that. Some of them have valves in them and some of them don't. So Venules veins. So veins are considered afferent vessels because they're carrying blood back toward the heart, and they contain a disproportionate amount of blood at any given time, because blood is kind of not on its in a hurry on its way back. So you have about 3 times as many veins as you do arteries, and there's a lot more variation in veins than there is in arteries. They have the same 3 tunics, but in, of course, different thicknesses. They don't have as much elasticity because they don't actually need it. And many veins have valves in them. And the job of a valve is, if something is going, for instance, against gravity and it doesn't have the pressure that an artery does, then it can try to flow backwards. But if you've got a valve, the valve will prevent backflow. So let me show you how valves kind of work. So here's how a valve works. So these are one-way valves, and the one-way valves promote unidirectional flow of blood even at low pressure. And this guy right here, as you contract your skeletal muscles, it pushes on the veins. And since it pushes on the veins and the blood can't go backward, it goes forward, forward, forward. The same thing happens with your respiratory system. So this is called the skeletal muscle pump, and it helps with venous return of blood through veins back to the heart. And then this is called the respiratory pump, and the respiratory pump actually changes pressure. So in this picture, it's showing you, okay, so this is your diaphragm, and your inferior vena cava runs through your diaphragm. This is, of course, your thoracic cavity and your abdominal pelvic cavity. Well, what happens when you breathe in, this is breathing in, is your diaphragm pushes down. It was bell-shaped, and then it pushes down. As it pushes down, it increases pressure in the abdominal pelvic cavity, and it comparatively decreases pressure in the thoracic cavity. And when it does that, since all fluids, including blood, flow from high pressure to low pressure, it makes the pressure higher here, lower here. Every time you breathe in, you squirt blood up your inferior vena cava. So that's called the respiratory pump. That's the idea there. Sometimes these valves that we're seeing right here, they get kind of weak. See, I forgot to bring this up for you because I always forget something. And you can get varicose veins. Find them for you. Okay. Sometimes the valves can get really weak and you get these varicosities. It especially happens in the superficial ones because there's not as much skeletal muscle to squish the blood up. There's a genetic predisposition for this, and of course if you are standing and not walking a lot, you are more likely to get varicose veins. So there's some varicose veins for you, and they can get painful when they get big like that. And then a varicose vein that occurs in the anal cavity is called a hemorrhoid. And those occur when you are sitting an awful lot, which causes problems getting the blood back up. Also occur when you're pregnant because of all of the extra pressure up here, causing the blood to have a harder time coming back up. Okay, now a vein that is larger than other veins, and really thin-walled is often referred to as a sinus. And I know that you guys know some sinuses. You should know some that exist in the head, and you should know some that exist, or one that exists on the heart. So can you think of some sinuses? Large, thin-walled veins. You've got all of the dural venous sinuses, including the superior sagittal sinus, the straight sinus, the sigmoid sinus. Those are all called sinuses because they're big, wide veins. And then what's the one that runs around the back side of the heart and dumps into the right atrium? That is called the coronary sinus. Okay, so there's some examples of those. All right, so now let's get away from the general vessels and get into the specific vessels. And first we'll do the easy one, which is the pulmonary circulation. Come on. Okay, so the circulatory routes. So remember that if we're talking about pulmonary circulation, we are going to be going from the right atrium through the lungs back to, from the right ventricle through the lungs back to the left atrium. And the pulmonary circulation is way, way lower pressure than systemic circulation. And I always ask this question, but I'm going to try it again. And you guys aren't here, so I can't tell if you get it or not, so we'll do our best. What is it that accounts for the difference in pressure between pulmonary circulation and systemic circulation? Not why is it okay that pulmonary circulation is way lower pressure than systemic circulation, but what anatomically accounts for the difference in pressure. And I asked you guys this before, hopefully it's actually in there now. What anatomically here accounts for the difference in pressure between the pulmonary circulation, which is 25/10 on average. 25 is the pressure in the arteries immediately after ventricular contraction, and 10 millimeters of mercury is the residual pressure right before the next ventricular contraction, so 25/10. Whereas with systemic circulation, it's on average 120 right after ventricular contraction, and then 80 right before the next one. So one last time, what is it anatomically that is responsible for the difference in those pressures? And hopefully you know that it is the difference in thickness between the left ventricle and the right ventricle. The left ventricle is much thicker and can generate more pressure. So the general path, if you were looking at pulmonary circulation, is from the left ventricle to the lungs back to the right atrium. So we are going to leave, so I did it wrong, sorry, I'm spacey today, from pulmonary circulation, I was testing you, from the right ventricle to the lungs back to the left atrium. So just get that right because I got it wrong. So if we are talking about pulmonary circulation, we are going to go from the right ventricle to the lungs, right? And then we are going to go back into the left atrium, even though Melanie could not say it right today. Allergy pills is what I'm blaming. Okay, so as the blood leaves the pulmonary circulation, or goes into pulmonary circulation, first it's going to leave the right ventricle, and it's going to go through a valve that they didn't show here. So what is the valve that is between the right ventricle and the pulmonary trunk? It is the pulmonary semilunar valve. So the pulmonary semilunar valve goes here. Then I go into the pulmonary trunk, okay, out this way. And then I'm going to go into the right and left pulmonary arteries. And then I'm going to go into the lungs. Now, these aren't shown here, but I will go ahead and tell you, even though I wouldn't ask you off of a figure, I just want you to kind of understand that since there are 1, 2, 3 right lobes on the lung, the right lung, and only 2 on the left, don't be surprised that when you go left-right pulmonary arteries, you branch into 3 right lobar arteries to go to the 3 right lobes, and 2 left lobar arteries, and then you start branching into smaller and smaller arteries and arterioles until you get to the capillary beds of the lungs. Now the capillary beds of the lungs are really weird looking. So let's figure out what's going on with those. So where I want you to see is that we went through these guys right here, right? And we're taking deoxygenated blood to the lungs. And then we are going to bring that deoxygenated blood, let's follow where we are, deoxygenated blood into the capillary beds of the lungs. The capillary beds of the lungs are weird though, because they separate into these mini capillary beds that each wrap around an alveolus. And this alveolus right here, these guys right here look like grape bundles. And what those are is those are the locations where air came from the trachea to the bronchi to the bronchioles all the way into the lungs. And they're just made of a single layer of simple squamous epithelium, and they're going to have air in them. Okay, so bring it to the capillary beds of the lungs that surround your alveolar air sac. And then what happens, of course, is as the blood goes by the alveolar air sac, so watch just this one, let's do this one. As the blood goes by, it comes in deoxygenated. It goes by this really, really thin alveolar sac, and the capillaries themselves are really thin, and it gets oxygenated as it goes by. And then it goes back into tiny little branches that are going to ultimately lead to the pulmonary veins. So it would go into little pulmonary venules and then bigger veins, and then eventually it would lead back to the right and left pulmonary veins with nice oxygenated blood that would dump into the left atrium, okay? And that would bring that oxygenated blood right back into the heart. And then, of course, the heart would start pumping it out into systemic circulation. Okay, so now I'm going to go back into the left atrium. After I'm in the left atrium, I will go through the bicuspid or mitral valve, and I'll go back into the left ventricle, okay? And once I'm in the left ventricle, when I leave, I will be in systemic circulation. So let's look at systemic circulation. Let's get rid of these. Okay. So now I am going to leave here, and I'm going to go into systemic circulation. So left ventricle and out, back to the right atrium is systemic circulation. So let's look at big picture understanding of systemic circulation. So first thing about systemic circulation is just how you talk about it. So of course most of the arteries and veins that you will learn are systemic arteries and veins, because there's way more of them, because of course there's way more systemic organs than just the one pair of lungs that's part of pulmonary circulation. So when you talk and do blood traces, which you're going to be working on this week, I want you to try, if you can, to get away from just saying, oh, okay, that's the femoral artery. I want you to always think about which direction blood is flowing through the femoral artery and what it is supplying. Because arteries supply, and they are going to supply blood, nutrients, oxygen, right, to whatever capillary bed they're going to. So think about directionality when you're doing that. But veins don't supply. Veins drain. So if I were, for instance, talking about that femoral vein, I would be like, okay, that femoral vein is draining blood from, for instance, the muscles of the thigh back into systemic circulation. Whereas this femoral artery right here come on. Why can I not get that? This femoral artery right here is supplying. Okay, finally got it. Okay, so this femoral artery is going to be supplying. So the blood's going that direction through the femoral artery. It's going the other direction through the femoral vein. So systemic arteries are going to be relatively high pressure, 120/80. 120 is the pressure in many systemic arteries immediately after ventricular contraction. 80 millimeters of mercury is the pressure in systemic arteries immediately before the next a ventricular contraction. The left ventricle works about 5 times as hard, not more frequently, but just uses up a whole bunch more nutrients for all of that myocardium that's in there. And arteries are going to, again, supply blood to capillary beds. You follow an artery from the heart to the capillary bed. And the systemic arteries, all of these that you're seeing right here, systemic arteries flow from what ventricle? All systemic arteries ultimately can be tracked back to what ventricle? It's the left ventricle. And they're going to go to body capillary beds with the exception of the lungs. And all systemic arteries are going to branch from the aorta. So if you're doing a trace, and I think all of your traces have systemic circulation in them, so every single straight trace is going to go through the aorta. And of course there are 3 branches of the aorta, and let's look at them in a little bit more detail. You have the ascending aorta, and they're named for which direction the blood is going through them. You have the aortic arch right here, and then you have the descending aorta. So ascending aortic arch and then descending aorta. Okay, so let's talk just briefly about veins overall, and then we'll get into the specifics. Okay, so if we are talking about veins, remember that you have way more veins than you do arteries. Okay, so there's lots more variation in veins. Veins vary from person to person, so if you were still as lucky as we were before and we're looking at cadavers, if the picture said that it branched right there, that's just a suggestion. It can branch anywhere it damn well pleases. It might branch up here into these two. It might branch down here. So what you will always do when you're actually working in clinical things is not say, hey, this is what it's supposed to look like, and only expect it to look that way, especially with veins. There's lots more variability. There's 3 times as many, and they branch anywhere they damn well please. But there's a way to not get confused by it. Because generally speaking, if you are watching it go through the arm, it's going to be called the brachial. The ones that are running on this side are going to be the radial, and these are the ulnar. So there's a lot of logic to it. So lots more variation in patterns, location, and branching. There are 3 times as many. Generally, although not always, veins will run along with their artery partner. This is especially true in the limbs. It's not very true in the abdominopelvic cavity, but when it occurs, it's helpful. And every single systemic vein is going to end up back in either the inferior vena cava if it's coming from below the heart, the superior vena cava if it's coming from above the heart. And although it's not visible here, if it's coming from the heart itself, it's going to end up in the coronary sinus. And then all 3 of those vessels, remind you, all 3 of those vessels are going to dump back into the right atrium. So all of them are going to dump back into the right atrium. So this is right atrium right there. Okay, so let's do a little bit of the blood flow. Okay. All right, so we're going to talk about systemic regional blood flow, and basically the way that I teach this is I'm going to basically talk about all of the major vessels as they come off of the aorta, okay? So since we're talking about systemic circulation, we'll start with systemic arterial circulation. And when we're talking about systemic arterial circulation, we start at the aorta. This is the ascending aorta. This is the aortic arch, and this is the descending aorta. So let's get stuff off of those locations. So first thing, the ascending aorta gives rise to arteries that supply the heart itself. And you've already learned this, but I want to remind you that you're going to have to go back and revisit it, because on the 4th test, and that this material is part of the 4th test, you have all of these vessels. But the ascending aorta gives rise to the arterial supply that feeds the heart muscle itself via the left and right coronary arteries. You've already learned them, but I want to just remind you that they exist, and I want you to write them down here. You also had to do this for your take-home question, so you should be prepared for it. I'm not going to go through the whole thing for you again, but I want to remind you of this picture, which is where it is, okay? So this figure is 22-9. So the first and only real branches, I mean, there's a smaller one, but you don't learn it, of the ascending aorta are the right and left coronary arteries. So here's my left coronary artery, yeah, and it's taking blood from there to there. Here's my right coronary artery right there, taking blood from the aorta here, okay, and then it's going to give rise to these other arteries. So what these are going to do is they are going to deliver blood from through those arteries and then arterioles and then to all of these capillaries within the heart wall itself, okay, and then they are going to deliver oxygen and nutrients to the heart wall itself, and then you're going to have to bring that used blood back. And the way that you are going to do that is you're going to bring it back up these vessels, the great, middle, and small cardiac veins that all dump into the coronary sinus, and then the coronary sinus dumps back into the right atrium. So this is all the first part of systemic circulation, because the heart is a systemic organ that needs blood for itself. Okay. So let me get all those out of the way. So you already have an assignment to do that. You don't have to do the whole thing. You have to do blood that goes to the anterior wall of the left ventricle, the myocardium in the anterior wall of the left ventricle. Okay. And then next, I am going to go to the aortic arch. So let me go down to my aortic arch. And the aortic arch has 3 big branches, although we did have one cadaver that had 4. That was very confusing to all of us, but variability exists. So the 3 branches of the aorta, or the aortic arch, are this guy right here. This is the Brachiocephalic artery. I'll tell you why it's called that in just a second. This guy right here, which is the left common carotid artery, and then this guy right here, which is the left subclavian. So here's the deal, guys. The reason that you have these 3 arches is that the aorta in almost every person arches over to the left side. So it goes to the left side. So stick with me for a second. That means that if I want to deliver blood to the left side of the head and to the left arm, I can go straight off of the aorta because it's already over there. So what's going to happen is this guy is going to give rise to, this is the aortic arch, this is the left side. It's going to give rise to the left common carotid, which will give most of the blood supply to the head, and then it's going to give rise to the left subclavian, and the left subclavian will give most of the blood supply to the left arm. But what about the right side? Well, the right side, so I arched over to the left side, but I still have to get blood over to the right side. So how do I do that? I have to make a little jog over to the right side, which gives the right arterial circulation one extra little vessel. So this vessel right here is called the Brachiocephalic Trunk, or sometimes just the Brachiocephalic Artery, and you really only have one on the right side because you didn't need one on the left side because you were already over there. And it's called brachiocephalic because it's going to give blood to the right arm, brachio, and head, cephalic, so brachiocephalic trunk, okay? The brachiocephalic trunk will, if you give it a minute, branch into the right common carotid artery and the right subclavian artery. After it does that, after you are in the subclavians or the common carotids, the arterial flow, left and right side, is identical. The only difference is that, generally speaking, from the aortic arch, if you're going over to the right side of the arm or the head, you're going to need this brachiocephalic trunk or artery first. and that's just because the aorta arches left. Okay, so that's what we saw right here. The brachiocephalic artery or trunk supplies the right side of the head and the neck, and it gives rise to the right common carotid and the right subclavian, called subclavian, by the way, because it goes under the clavicle, hence subclavian. But the aortic arch gives direct blood supply to the left common carotid and the left subclavian. Okay? All right, so I think the last thing that I'm going to do today is just get blood to the head and back. Okay? And then I'll pick up the next one after you guys take your test. Okay, so arterioles, blood supply to the head. All right, so from the picture that we just looked at, can you kind of tell that most of the blood supply to the head is going to be through these two common carotid arteries? So let's get those out of the way and then sort of orient you. So I'm going to show you a different picture. It's right here, okay? And what this one is doing is it's showing you a right lateral view, and it's going to get blood supply to the head. This is not as hard as it looks. It's actually not that bad. So how do you get blood supply? Most of the blood supply to the head comes from the right and left common carotids. And the common carotids, do you see? This is on the right side. So since we're on the right side, the blood would have been in the aortic arch, and then to get here, oops, that's not what I want. To get here, the blood would have had to come up the brachiocephalic artery. If I was on the left side, I don't need a brachiocephalic artery. And then next it's going to go up the right common carotid artery. We're not going to do this one quite yet. And then the common carotids are called common because, generally speaking, when you run into a common vessel, it is going to split. So the common iliacs will split into internal and external iliacs. The common carotids are going to split into internal and external. It's not always internal and external for every common, but that's not unusual. Okay, so now we are going to split into, the blood is still flowing up toward the head, by the way. We are going to split into the external carotid artery, and the external carotid artery right here, you can see where it's going, right? It's going here. And it's going to supply most of the facial structures and the bones of the skull. So now I'm delivering systemic, highly oxygenated blood to my facial structures on the outside and bone. But then this guy right here, do you see him? That guy right there? That is the internal carotid artery. And the internal carotid artery is going to go inside the skull, and it's going to go through, ding, 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 the carotid canal of the temporal bone. And it's going to serve, the left and right internal carotid arteries are going to serve about 75% of the blood supply to the brain, go through that pair of arteries, okay? But there is a little bit more that we have to pick up because we've got 25% more blood that we actually need to go in there. So the last little bit of the blood supply to the brain that we are concerned with actually comes from here. So remember that now we're on the right side. So this was the brachiocephalic artery or trunk. This was the right common carotid. And then this is the subclavian. Do you see this little branch right here from the subclavian that's going up the transverse foramina of the cervical vertebra? That is called a vertebral artery. And the vertebral arteries run up the transverse foramina of the cervical vertebrae, those extra holes on the side. And what they are going to do is run up the transverse foramina, and then they are going to dive anterior to the spinal cord and run up the foramen magnum. The pair of those, the two vertebral arteries, are going to give the rest of the blood supply to the brain, that last 25% of the blood supply to the brain. So let me show you how that works. So they are going to do that. So this is going to totally flip this picture for you, but it's an important one. That is right here. So we're going to look at the bottom side of the brain with these vessels that are delivering blood to the bottom side of the brain. So now let's figure out where the heck we were so that we can make sure we orient ourselves. All right, so now we are delivering the last 20, we're delivering all of the blood supply to the brain, sorry. And so now let's figure out where we were. This is one of your internal carotids. This is the other internal carotid artery. They are going to come up and deliver blood right around the hypothalamus and pituitary gland. Okay, let's see. That one's, yeah, this one's left one and that one's right one. So 75% of the blood comes right there. The other 25% of the blood comes through these two. These two are your right and left vertebral. So they were going up the transverse foramina of the cervical vertebra, and then they dive anterior and then go up the foramen magnum. And that's those two right there, the vertebral vertebra. And then, by the way, the vertebral artery. Vertebral arteries are going to come together. I don't make you learn the name of this one, but this little guy right here is called the basilar artery. That basilar artery runs in the cleavage of the pons, and then it runs up and it makes this circle, right? And this circle is a bunch of redundant circulation for the brain, because if this one got blocked right here, if that guy got blocked, I could still get blood all the way around. This is a fantastic example of that thing that I told you guys about called collateral circulation, which is redundant circulation to make sure that you can still get blood through it. This right here, this circle is called the cerebral arterial circle, or the circle of Willis, and it's actually what I'm wearing on my necklace right now. You see it? Circle of Willis. And if I were in class with you, I would tell you that the circle of Willis is actually why I am alive, because a few years ago, one of my vertebral arteries busted a hole and started dumping blood straight into a vein. If I didn't have all of this redundant circulation, I would have lost blood flow to the back part of my brain. So, but I didn't. The vertebral arteries, we've got those. And remember, the vertebral arteries came off of the subclavian artery, vertebrals. And collectively, the two vertebral arteries and the two internal carotid arteries give all of the blood supply to the brain, almost all of the blood supply to the brain via the circle of Willis, or the cerebral arterial circle. Okay, and then the last thing I'm going to do today is just how do you get all that blood back from the brain back into systemic venous circulation. And so let's look back at this picture for a second and say, okay, now we're going to try to get the blood back. Okay, and so let's get rid of a bunch of these guys. Come here. Okay, so how do I get blood back from the head? Blood is going to go back from the outside of the head. So watch here and you'll be able to see these really well. So blood is going to go from the outside of the head through this vessel right here. Do you see this guy right here? This guy right here is the external jugular vein. How can you tell it's the external one? Because it's actually draining external structures. So the external jugular vein, it drains the facial muscles and the scalp that were supplied by the external carotid artery. So that's the external jugular vein. Notice, though, it actually dumps into the subclavian vein. Okay, external jugular dumps into the subclavian vein. And then these vertebral veins right here, vertebral veins, vertebral arteries. Vertebral veins also dump into the subclavian veins. So we got the outside of the head, we got 25% of the inside of the head. And then now what we are going to go back to do is figure out, okay, all of those dural venous sinuses, but just let me simplify it for you for a second before we get into the nitty-gritty of the dural venous sinuses. All of the blood from inside of the brain is ultimately going to end up in this vessel, and that vessel is the internal jugular vein that is going to go through the jugular foramen. But what we're going to have to do is we're going to have to look inside the head a little bit to find all of those dural venous sinuses. Ultimately, though, they're all going to dump into the internal jugular vein. So let's show you a couple pictures, and then I've got an animation for you guys that just sort of puts it all together. Okay, so let's see where these are going to go. So the superior sagittal sinus, you see that guy right there? The blood is going to go through the superior sagittal sinus, that direction. This is the inferior sagittal sinus. The blood is going to go that direction. Okay. This is the straight sinus, right? Going to come around here. All of it is going to start to collect in the sigmoid sinus, going down. And then the sigmoid sinus is going to dump into the internal jugular vein. There's a couple of these back here that you can't see super well from that view. Got another picture for you that came from a different textbook. Oh, this one's cool too. This just shows an inferior view of the brain. Kind of like this one. It's got more vessels than you need to know on it, but you can see the superior sagittal sinus coming into the confluence of sinuses, right? You can see the transverse sinus that would be going in that way. So this one's going in that way, okay. You can see the sigmoid sinus right here, okay. Oops, that one should be going the other direction. Why is that one that way? Oh, confluence of sinuses. So you can see the sigmoid sinus here that's coming in that direction, okay. And then what are they all dumping into? They're all dumping into the internal jugular vein. So let me show you an animation that shows you the same thing. And then we will be done for today. All right, ready? You get out of the way. Okay, make sure you can hear this.
Venous blood from the brain drains into a system of valveless channels called dural venous sinuses. These sinuses are located at the margins of dural folds where the periosteal and meningeal layers of dura mater separate. Along the midline, two sinuses that receive venous blood from the brain and meninges are the superior sagittal sinus and inferior sagittal sinus. These lie in the superior and inferior margins of the dural fold, known as falx cerebri. Blood in the superior sagittal sinus flows posteriorly to the confluence of sinuses. Here it unites with the straight sinus and occipital sinus. The straight sinus is formed by the inferior sagittal sinus and the great cerebral vein. The paired transverse sinuses extend laterally from the confluence of sinuses and run horizontally along the internal margin of the occipital bone. The transverse sinuses continue as the S-shaped sigmoid sinuses. The sigmoid sinuses enter their respective jugular foramina to become the internal jugular veins of the neck.
Okay. See if I can figure out how to make that go down.